Hello, this is Mrs. Scoville, and today we're going to be learning about DNA structure. For today's lesson, we have 3 learning objectives. The first is to be able to identify DNA base pairing rules, to describe the structure of DNA, and we're going to get a little history lesson on that one too, and to be able to write out a complementary strand of DNA, and that's one we're going to continue to work on. Okay, so for starters, we need to talk about what DNA is. I'm sure you've heard those letters, DNA, but you may not know that it stands for a very long word. So DNA is deoxyribonucleic acid. So you can see the D, the N, and the A, so DNA. And you may already know that this is the genetic material that's present in basically any living organism that we are currently aware of. So we say it's hereditary because it's passed on from one generation to another. Okay, so this is something that's in all living organisms. You get a little preview of the structure here as well. So DNA is composed of nucleotides. So DNA was one of our examples of a nucleic acid, and that was one of our 4 major macromolecules, the others being proteins, carbohydrates, and lipids. So again, DNA is a nucleic acid. And since DNA is a nucleic acid, it is going to be a polymer that's made up of nucleotides, or building block of nucleic acids. And as a reminder, this is a little bit of review, nucleotides have 3 parts. So the first part is going to be a nitrogenous base, and nitrogenous, that just means it contains the element nitrogen. It will also contain a 5-carbon sugar, and finally it's going to have a phosphate group. So we're going to get into more detail here with the 2 different types of bases, but you can see basic structure. We've got our 5-carbon sugar, and it looks like a little pentagon or pentagram here, and the phosphate group, and then we're going to have one of 2 kinds of bases attached, and again that is going to contain nitrogen. So DNA, I said already that it was in basically all living organisms. That includes prokaryotes like our bacteria. So as a reminder, in prokaryotic cells we're going to find DNA in the cytoplasm. It's not enclosed in a nucleus, and in eukaryotic cells it will be inside the nucleus. And also this is a little bit of review as well. So remember, in our eukaryotic cells, in both plants and animals, there are also small amounts of DNA in the mitochondria and in the chloroplast if it's a plant cell. As we've seen recently, thinking about mitosis in our nucleus, in our eukaryotic cells, DNA is tightly packed to form chromosomes. Remember, in our prokaryotic cell, it's not going to look the same. Okay, so now we can see it here. I was kind of alluding to this, that in our prokaryotic cell, it's not in a nucleus, it's just there. So in either case, DNA is going to be double-stranded. So again, that just means exactly what it sounds like, that it has 2 strands. And these 2 strands, or 2 kind of strings, are going to be connected to each other. In our prokaryotic cells, again, the DNA is going to basically form one continuous long looping chromosome, and it is basically free-floating in the cytoplasm of the prokaryote. It is not in the nucleus. And in our eukaryotic cells, so plants or animal, it's going to form this spiral called a double helix. And this is probably what you're more familiar with when you think about DNA. So we're going to get a bit more into the structure of this, but I do want to point out a couple of key features here. One, you can see that nucleotide we already mentioned, and also the sugar and the phosphate in the nucleotide are going to make up what is called the backbone of the DNA. Okay, so you're probably thinking at this point, how on earth did anyone come up with this structure? So as early or as late, depending on how you think about science, as the 1940s, DNA was suspected to be the molecule of life. It was not known to be, still in the 1940s, it was not known to be kind of our hereditary information because people thought it was too simple. Actually, prior to this point, a lot of people thought it was more likely that protein was going to be the molecule of life that maintained life, because DNA just was too simple. It had just these 4 bases, and it was awfully simple. But people really thought this might be it. So a lot of scientists around this time were very interested in trying to figure out the structure of DNA. And in 1953, 2 gentlemen, James Watson and Francis Crick, were using a lot of stick and ball models to come up with a helix structure. And they were able to come up, even just using these stick and ball models, with a pretty successful helix structure but this needed to be confirmed scientifically. So Rosalind Franklin is the main person that's credited with this picture right here, which is showing an X-ray crystallography photograph. And this may not look like a lot, but here, and it's not X-ray crystallography because this looks like an X, that's just a technique that's used. But the fact that this actually formed an X in the picture indicated to them that it was a two-stranded helix. So this was used as evidence that the structure is what we see it as today. Okay, little music there. So now we're going to look a little more at the actual double helix of the molecule. So again, just a reminder, DNA has a double helix, and this means that the two strands are twisted together, as we kind of saw in those previous pictures. Let's look now at just one strand of DNA. Let's break it down a little bit more. So again, the backbone I kind of alluded to it before, but let's make it really clear now. The backbone of DNA is going to be made up of phosphates, and DNA is deoxyribonucleic acid because the 5-carbon sugar that it's made up of is called deoxyribose. So our backbone is sugar, the sugar being deoxyribose, and phosphate joined together. So that's going to form the main outer backbone structure of DNA. And what look like little teeth here coming off, those are going to be the nitrogenous bases. So if you kind of look here at an angle, that's our nucleotide. We've got the 5-carbon sugar, the phosphate, and the nitrogenous base attached. All those joined together, that forms our polymer, which is DNA. Of course, right now we're only looking at one strand. And you can see right there I said the nucleotide again. So strand of DNA, it's the polymer of nucleotides, absolutely. And just a note, one strand of DNA, nucleotides are quite small. One strand of DNA is going to have millions of nucleotides. Okay, so now let's get more into those nitrogenous bases. There are two kinds of bases that you can find in DNA. The first is called a pyrimidine, and those are the ones that are just going to have a single ring. So just right here, one ring. The other type are called purines, and these are double ring bases. So I can't really think of a great way to help you remember these. I actually just remember that they're kind of opposite from what I think. So pyrimidine, name is more complex, but they're actually simpler. Purines, the name is simpler, but they're more complex. So if you can think of some other way for yourself to remember it, that's great, but that's how I remember it. So again, pyrimidines, single ring, purines, double ring. There are two pyrimidines. They're named thymine and cytosine. And again, they're going to have one ring. And when I talk about a ring, I'm meaning a ring made up of carbon and nitrogen. So structure of thymine, structure of cytosine, you can see they're very similar, carbon and nitrogen in the ring. The main difference just comes to what's bound to the ring. So in cytosine, there's a nitrogen in place of an oxygen, and that's really the only difference. Adenine and guanine, on the other hand, are going to be purines. And again, purines, shorter, they're actually more complex, so double ring structure. And so here again, adenine and guanine, they're purines, so you see a double ring. And notice one ring is 6 and one is 5. They're directly attached to each other. Again, basically one element difference, an oxygen versus a nitrogen. Those differences are going to be important though, because that's going to lead to differences in bonding. So if we look at two-stranded DNA, we need to remember that they're basically going to fit together. So we've got one strand here and another strand here, and notice they almost fit together like a zipper, like teeth in a zipper, or sometimes people refer to DNA as a ladder, almost like these are rungs in a ladder. But I do want to point out to you here that if we look at how these two strands are joined together, you can see it very well looking at the deoxyribose sugar. These strands are actually running in opposite directions of each other, and this is going to be quite important later when we get into DNA replication. So overall, though, the question is, why are these actually sticking together? So especially if you look at these two, they don't really look like they necessarily fit. So the bases, as it turns out, are actually attracted to each other, and this is going to be due to hydrogen bonding. So hydrogen bonds, we haven't talked about them in a long time, but they're very weak bonds. So by themselves, they don't do very much, because there are millions of nucleotides in a strand of DNA. There ends up being millions and millions and millions of these weak hydrogen bonds in one single molecule of double-stranded DNA. Overall, this actually leads to a very stable, strong molecule. Here, you can see in this picture, this is showing cytosine and guanine. So cytosine's here, guanine's here, and you can see that there are actually 3 hydrogen bonds that are going to form between guanine and cytosine. So again, cytosine, it's our pyrimidine, so it's simpler, and up here this would be guanine, and it's actually a purine, so it's more complex. Okay, so a little bit more here. Little bit of pairing, so I kind of said already here, we've got cytosine and guanine. Well, cytosine is always going to pair with guanine. That wasn't a fluke. And adenine is always going to pair with thymine. So we kind of say these are our DNA base pairing rules. So here we've got adenine bonded to thymine, and adenine here, it's again going to be more complex. So since it's actually more complex looking, that means it's a purine, and thymine is right here, it's simpler, so it's actually a pyrimidine. So again, the names are kind of opposite in complexity. Notice here, adenine and thymine only form 2 hydrogen bonds between them. So this rule that I just said, adenine always is going to bind with thymine, and cytosine and guanine are going to join together, is known as Chargraff or Cargraff's rule, and it's named after, well, Chargraff. So this gentleman came up with this rule, and this is actually before they came up with the whole structure of DNA. They determined that in DNA they were always in equal amounts. So you always had equal amounts of adenine and thymine to each other, and cytosine and guanine. So when they unraveled the structure, this made a lot of sense because these 2 always paired together and these 2 always paired together. That video we'll get to in just a second. Because of Chargraff's rule, though, we can always determine the other strand if we're given half of the DNA, because A always pairs with T, C always pairs with G. So the first example here, you can see we have a sequence ATG, CTT, GCA. Those are just shorthand for adenine, thymine, cytosine, and guanine. So adenine always pairs with thymine, thymine, adenine, so you can always just do the opposite when you're given one. So again, you can see it's just the opposite. So G with C, C goes G, A goes T. Okay, same thing here. Doesn't matter what you start with, you can always figure out the opposite. Okay, so now I'm going to back up here, and we're just going to watch this short little video.
Two DNA strands intertwine to form a double helix. Each strand has a backbone composed of phosphates and sugars to which the bases are attached. The bases form the core outside. Grooves between the backbones or protein DNA contacts are made. The DNA backbone is assembled from repeating deoxyribose sugar units that are linked through phosphate groups. Each phosphate carries a negative charge, making the entire DNA backbone highly charged and polar. A cyclic base is attached to each sugar. The bases are planar and extend out perpendicular to the path of the backbone. Pyrimidine bases are composed of 1 ring, and purine bases of 2 rings. Adjacent bases are aligned so that their planar rings stack on top of one another. Base stacking contributes significantly to the stability of the double helix. In a double helix, each base on one strand is paired to a base on the other strand that lies in the same plane. In these base pairing interactions, guanine always pairs with cytosine, and thymine with adenine. A GC pair is stabilized by 3 hydrogen bonds formed between amino and carbonyl groups that project from the bases.
So the 3 bonds actually makes the GC bond more stable than an AT bond.
In contrast, an AT pair is stabilized by 2 hydrogen bonds.
This means DNA with more GC bonds is more stable.
The specificity of base pairing, that is C always pairing with G and A always pairing with T, ensures that the 2 strands are complementary. This is important for DNA replication,
which we will get into at a later time. So that is all for today's lesson about DNA structure. Make sure you write your complete summary.